Hello guys, you're welcome to my YouTube channel, and thanks for coming back again. If you are already the subscriber of this channel, and if you are new here, endeavor to click on the subscribe button, and I hope you like that, because we're gonna be dropping good content as you come here, you get them. So in this video today, we want to look at the WAEC 2023 biology practical, and we want to talk about some specimens, and then this um going to be very useful for the upcoming examination. So we are going to describe our specimen with letters just like as usual. So we have the specimen A to start with. So let's take a look at specimen A. This is a catfish egg, the fresh one. So looking at this, what question could come out of this? Catfish egg, the fresh catfish egg. So, and we have a scientific name for this particular fish. It is called Clarias gariepinus. So this is the name of this fish. So let us describe the egg and how it looks like. The egg channel of catfish, they are large, and they're about maybe 2.4 to 3 millimeter in diameter. So that is the size of each egg. And mind you, the egg consists of balls of uh, cells in it, which are usually something like oval or close to spherical shape, but not actually perfectly spherical. So now we have other properties of it, and we know that it is adhesive when touched, so which means that it has the property of an adhesive material. Then it is usually laid in a large mass, so which means this egg is laid in large mass. So a large quantity of the eggs are laid at once. Then I've said this earlier, they are small, spherical like in shape. So another thing is that they are transparent and progress from pale yellow to orange color as they mature. So when the egg are laid, you can see them in their transparent form, but as they mature, the color begins to change to the present color you are seeing now, which is orange color. So every egg of the catfish is always mature at that orange color. So that is what it implies. So now looking at other properties, we look at the time it takes the egg to hatch. So it depends on the condition where the egg is being located. So this might be the possible questions you'll be asked. So the time it takes the catfish egg to hatch depends on the temperature. I've said that. So, and let's say the temperature should be between 21 to 29 degrees Celsius. So, and the time for spawn to hatch is maybe 5 to 10 days. So once the temperature is favorable, it's going to get hatched between 5 to 10 days. So this is, as you can see those in pictures in the diagram, so at least it has shown the different stages of the life cycle of the fish. So starting from the egg. So let's move to the next specimen. We have the egg of a fowl. The egg of a fowl. Now, the fowl egg is common. We can get that everywhere. I think we are familiar with that. So we know it is made up of a shell, and inside the shell you have the liquid, which is divided into different layers. We have the egg white, that is the white part of the egg, and when you look at the solution with the liquid content of the shell, you would discover that some are lighter as you go towards the yolk, that's center of the egg, which is actually now yellow, pale yellow, or let's say yellow color. Is intense yellow color actually. So you have the thickness and more thickness till you get to the yolk side. So this is one of the property of the egg. So the white part of the egg is called the albumen. It is a clear liquid, so it turns opaque by the time you cook it. If you cook that egg, it will become white. The colorless, which is called the albumen, will become white and opaque after it has been cooked. Then the yolk, which is odd, which is yellow at first, will become pale yellow when it is cooked. So other things we need to know about it is that they are hatched in 21 days after they have been covered by the female fowl, which is the hen. So, and they are edible because they contain protein. So let's look at the diagram that shows the label. We can see the shell, then we can see the thin albumen layer, then we have the thick albumen layer. As I said, we have the layer that is close to the shell is a little bit not as thick as the one that is at the middle. Then we have the yolk. So then in the yolk, you can see the germinal disc or the blast disc. That is what it is called. So then we have the layer which divides the yolk and the what and the thick albumen. That area is okay. Let's call that is the yolk. Actually, it is called vitelline, the yolk membrane, vitelline membrane actually. So then you can see some two set of white tissue there. They call them the air space. Okay, that is calaza. You can see the calaza there. So then we have airspace, which is that is the normal propagation of the egg. So looking at the egg that way, it is what it is longitudinal view of the egg. So not the normal way we do cut it. We do cut it in the other way around. But I think as you can see there, it was divided or it was drawn in the diagram in the longitudinal section. So let's look at specimen C. Specimen C is the uterus. This is human uterus containing fetus, as you can see in that diagram. So we know uterus is just, let's call it a bag or a body, and it has an hollow shape. So it is an hollow shaped organ. And what is the usefulness of uterus in man? It is actually responsible in varieties of function. Let's look at the functions. During gestation, that is pregnancy. Pregnancy is gestation. Then we have during menstruation and labor and delivery. So the uterus is very useful in each of these functions that we have mentioned. So now, an average uterus, we have the length of 8 centimeter, then it's going to be 5 centimeter across. So and then the thickness should be around 4 centimeter. So then other property is that let's just look at the uterus, the way it is divided. It is subdivided into 3 parts. So let's look at the first part. We name that the body, that is the actual body of the uterus. Then we have the cervix and the fundus. So we can assume that the body is the one that houses the fetus. So it is the major body, which with the sac-like body of the uterus. So then looking at the fundus, it helps to join the fallopian tube at either side, while the lower end will open into the vagina. So we can see that now from the diagram. Can we see where the fallopian tube is located? And we can see the diagram where we have the placenta, the uterus itself, and the fetus in it. Then we have umbilical cord, which is connected to the placenta. Then we have the amniotic fluid, that is the fluid that the baby, let's say, bathes in. Then we have the amnion. You can see that. So all that. Look at the cervix. So the cervix actually opens whenever menstruation occurs or during menstruation to allow blood flow out. Then it also helps in fertility, because if it does not open, the male reproductive cell, which is the sperm, would not find its way to the uterus. So uh, it opens during reproduction, so that the male sex cell rather, could have the access into the womb in order to fertilize the egg in the fallopian tube. So that is that. Then we have other advantages of it. It helps in fertility. I've said that. It connects the vagina to the uterus. Then that is, I'm talking about the cervix. Now what other thing is the fetus? You might be asked question about the fetus. The fetus is not an embryo. So an embryo is this very early stage of pregnancy. So when it grows for about, that is after 10 weeks, then we cannot call it an embryo again. So it becomes a fetus. So that is what we have on specimen C. So let's look at specimen D. So specimen D is the ovary of Pride of Barbados. So we know that we have a plant called Pride of Barbados. So we have specimen D, which is the Pride of Barbados, the ovary of Pride of Barbados. So this ovary, we know that ovary is a reproductive organ of that particular plant. So the plants will actually go for a kind of pollination. So, and that is a sexual reproduction. So where the seed the reproductive cells is being transferred. So, and then pollens are transferred from the anther to the stigma and all that. So we can state some properties about it, like it is positioned above the attachment point of the other floral plants such as sepal, petals, and stamen. So we know what that means. So the position of the oval is what we're talking about. Just look at it. So it is placed on the sepal, and you can see it is placed above also the petal and the stamen according to the diagram over there. So, and that is one property. It is a superior ovary. It is called a superior ovary because it contains the male and it contains the two cells in it for reproduction. It takes place, it reproduces itself by the transfer of seed from anther to stigma. So that is okay. The little we have about it. You can be asked to draw it. That is diagram. You can actually sketch it out yourself to see and look at the labels. Can you see the petals is in red color, the sepal is in green color, then you have the pedicel and the receptacle. Then we have the style, the ovary, the oval, the stigma, anther, and all that. They are all present in the diagram. So now let's look at specimen E, which is leaf of Pride of Barbados. So the leaf of Pride of Barbados is just the leaf of the previous ovary we've talked about. So it is just the leaf of that ovary we've talked about. So the leaf is a plant. It comes from the plant called Pride of Barbados. It is a shrub, or another name for shrub is small trees. Yeah. So it is legume family, belongs to the family of the legume. Then we can also say it is called the peacock flower because of the propagation of that the flower of Pride of Barbados. Then it has special petal. Then the special petal helps to lead insects to the nectar where they will suck the nectar and then aid pollination. So it is an insect pollinating plant. That is why we are saying that is the function of the what petals. It helps to lead insect into the nectar for them to suck and the plants get pollinated. So the leaf is a very good cause of a very good economic importance, so of which you can find out more about the leaves. So you'll be able to write something about that and probably make drawings. So as you can see in the diagram, we have the labels too over there. So now let's look at the specimen F, the panicum plant. The panicum plant is the guinea grass. So you can see the guinea grass over there in the picture in the video. And however, we also have another diagram showing the labels of the parts of the guinea grass. So it has oval, small, light green seeds. So there is seeds in the guinea grass. So it's often the seeds are often wrinkled. So they are always wrinkled. You can see. Then the lowermost branch of the seed heads are arranged in clusters. So let's look at the diagram. So when we look at the lowermost branches, that is the branches that is very close to the soil, used to have seed heads that are arranged in clusters. That means they are packed together. Then it has tufted grass with short creeping rhizomes. So there are rhizomes, and you can see that in the diagram, a very short grass with the creeping rhizomes. You can see the rhizome in the label. Then it is a perennial plant, which means, you know what that means. We have the annual, the biennial, and the perennial plant. So which means it can grow for a year longer than 3 years, longer than 2 years rather. So now it is actually used because cows and other grazing animals feed on this particular plant called guinea grass. They feed on it and they make use of guinea grass as their food. So it can be used as fodder for pastures and hay and silage. So there is one disadvantage about specimen F, which is guinea grass. The disadvantage is that they quickly spread on land. So in doing that, they actually become a weed plant. So that will be needed to remove from the plants, from the probably the farm area, if it is actually an area that is good for farming, because they will start to exploit other plants that are available in such surroundings. So that is a disadvantage they have. So, but one of the economic importance is the property which it has for it to be a form of food feed rather for animals, grazing animals. So now let's look at specimen G, that is cocoyam plant. Cocoyam plant is a plant with a greeny leaf that such leaf, I mean the leaf is having a big the leaf has the elephant here. They call the leaf elephant here due to the size, as you can see from the picture. So then the leaf is large shape. Then it's an herbaceous plant and a perennial plant. So herbaceous, which means it's an herb. Then it is grown primarily for edible roots. The root is very edible, and that root is the culm of cocoyam. So which is the next specimen we're going to talk about, specimen H. So the specimen H is the culm of cocoyam. So let's describe how this culm looks like. It is the edible root of cocoyam plant, like we have said, specimen G. Then it grows and germinates all in 8 months. Then the tuber is white and pink in color when you peel it. So when you peel the tuber, it actually shows the white, or let's say whitish pink color when you peel it. So, and because this plant has the property of being edible, so we're meant to talk about it and other importance of it. So the leaf can be used in preparation of herbs in local areas of Africa. So that is one of the use, and the scientific name for it is Colocasia esculenta. That is for the plant Colocasia esculenta. So you can be asked to draw it, either the culm of cocoyam. You can be asked to draw that. So all is still the same. So now the next specimen is the dry humus. The dry humus. Dry humus is let's say, let me tell you what humus is. As you can see from the diagram, it is a dark organic material that forms in the soil when plants and animal matters decay. When the dead plants and animal matter decay, so the resulting organic material formed on that soil is humus. So we are actually talking about the dry humus in specimen J. So we are actually going for specimen J now. So now this dry humus consists a lot of nutrients for plants to grow. Then because it is dry, there is lack of water, or it's no moist content, then it flakes when it is touched. It's crippled when it is touched, so it flakes off and crippled when it is touched. So these are the properties. You might be asked the importance. So it can be used as a very good um, nutrient source for plants in the soil. So that is for specimen G. Then we have specimen K, the moist humus, just opposite of dry humus. We've said a lot of things about it. So now this one contains moist. That means it contains a certain percentage of water. Then with this, there is a content of it which is humic acid is present, and this humic acid is useful for the soil pH. It increases the level of the soil pH, that is the moist one. Then it contains certain percentage of water. I've said that. Then actually is also good for planting. So now let's move to the next plants, the next specimen rather, which is orange, and that is citrus sinensis. Citrus sinensis, specimen L. So citrus sinensis is a citrus species and belongs to the family Rutaceae. Then, okay, let's describe it. It has a waxy skin. Then it contains carpels, and the carpels, when you look at the lateral view of orange, when you check the lateral view of orange in that diagram, you would see the segment it has been divided to. So the segments. So when you check the lateral view, you can see we have the segments there. So that is it. You can see it is divided into sections where you have the placenta and all that, where at the center then other sections are there. Then you might be asked to cut the orange in the longitudinal view. The longitudinal view is when you cut it sideways, abnormal from the way we used to cut orange. So but when you cut it the normal way orange is being sliced, then that is the cross-sectional view of the orange, or the lateral view. So then the longitudinal section of orange is when you cut it sideways in such a way that you're going to just see two segments. But by the time you cut it on lateral view or cross-sectional view, you would see that you have different segments, or let's call it segments actually. So now it also contains seed in the juiced filled vesicles. So we have vesicles in it, and these vesicles contains juice, and also in the vesicles you have some seed. So now let's talk about the shape of orange. It's generally spherical in shape. It is spherical in shape. Then other thing is it is made up of an outer peel and immediate fibrous layer. So which means from the exterior part of it, which is the waxy layer, immediately you peel that off, you're going to see a fibrous layer. Then after the fibrous layer, then you have a layer which is the juicy flesh and fibrous core. Then looking at the diagram, we can see the label. We have the epicap, the mesocap, the endocap. We have the placenta, the seed, and the succulent area. So that is what we have about oranges. And let's look at the method of reproduction in oranges. Is always asexual, and they do not bear naked seed. Their seed is eaten. So that is for specimen L, in case you might be asked any question on that. So now let's move to specimen M. That is Cocos nucifera. Cocos nucifera. It is actually a plant, or a fruit rather, a fruit that is edible for consumption. So we call that coconut. Then it is a round and elongated fruit of white, fibrous and oily meat covered by a thick brown hairy shell. So if you take a coconut, the outside is always very, very hard, which is the shell, and this shell looks hairy. Then the thick brown hairy part of it is the shell. Then inside the shell you have the oily meats. So and this oily meat is white and is edible. It also contains seed. Then it contains a 51.7% kernel. The kernel, the content is what we call the kernel. Then about 9.8% water, and then 38.5% shell. That is it. Looking at the diagram, we can see other labels on coconuts. We have the endosperm, we have the exocap, the outer layer, and we have the endocap. We have the roots, we have the fibrous husk. Then if you look at the complete one that has not been divided, we have the plugged spore, we have the functional spore, and we have the plugged spore. The plugged spore is actually two, and we have the functional spore. So that is the label and the diagram, so you can actually get one or two informations from them. Then they also use for cooking. The oil extract from it rather are used for cooking, and then for skincare purpose. So that is for coconuts, which is Cocos nucifera specimen N. So let's move to specimen M rather. So let's move to specimen N, which is the chili pepper. The chili pepper is a fruit called Capsicum annum. Capsicum annum rather. Capsicum annum. So it is a form of berry fruit, notable for their hot flavor. So which means they are hot in nature. Maybe probably when you chew them, if you know normal pepper get hot. So they contain seed, and this seed are attached to the wall of placenta, as you can see in that diagram. Contains stem which takes to attach to the plant. There is a stem where it takes to attach to the original plant it was plucked from. That is what you can see in the label, the stem. Then it does not bear naked seed. As you can see, if you did not cut it, you won't see the seed in it. So it does not bear naked seed. They are perfect flower because they contain stamen. So when you say perfect flower, that means they contain the stamen, which is the male, and the pistil, which is the female reproductive organs. So it means that they can perform what we call self-pollination. So they don't need any external agents such as insects to pollinate them. So they are self-pollinating plants or flower. So that is for chili pepper. So we can have you can look at the diagram of other label. You can see the other end from the stem is called the apex. Then you have the pepper wall, the placenta, the capsaicin glands, and the seeds. Then we have the calyx. So those are the various parts you can have. So you can try to make various drawings of all these specimens, so you can come out with a flying color.
Thank you for watching this video, and don't forget to subscribe to this channel. Thank you.